So, N. N is the number of different chromosomes. Fruit flies. N is the number of chromosomes that code and have different genes and genetic information on them. So we have 23 different chromosomes that have different information for coding for different genes. We have two sets of those, hence the 2N. One set comes from mom, one set from, there we go, okay. N is the number found in sperm or egg, okay? So, everybody understand? The N, so think of N as the number of genes, or number of chromosomes with different genes that are coding for different things, okay? All right, how many chromosomes do bacteria have? One, and it looks like a yummy. Germ cells. No, these are not the ones making you sick. All right, so germ cells being beginning. These are made through the process of meiosis, although it has an E in it. Why is that E silent? Who knows? All right, so germ cells made through meiosis. These are your sperm and egg. And the purpose of meiosis is to what? To make sex cells. To reduce the chromosomal number, yes. You can't pass on more than 23, so it's to reduce that chromosomal number down. We will get into the process of meiosis much more in depth when we get to genetics in January, probably. Histones. Turn to page 328 real quick. All right, histones. How long do we say our DNA is stretched out end to end?
17.
That's a lot of DNA. So how does our cells cram all that into a tiny little cell? Well, it coils up. Read A and B for us. Go, Jackson.
These are proteins that help DNA coil up, condense to form the chromosomes needed for division. When DNA is wrapped around these histones, the whole combined structure is referred to as a nucleosome.
Yummy. And they show that on 328 there in the electron micrograph at the top, says where it says nucleosome. And they've got like a little animated version of that as well. You can see the histones, they're just little proteins that look like a bowling ball with a fuse attached, or a bomb with a fuse attached maybe. And the DNA coils around that in the nucleosomes, and then it coils and then coils some more into supercoils. And you can see there's a lot of coiling taking place here until it gets into this chromosomal structure that we're familiar with. Okay? So, that helps it to coil up so that it fits in there and makes it easier for replication. Sister chromatids. Angela, read that for us.
A portion of the whole duplicated chromosome. This term refers to half the duplicated chromosome. Duplicated chromosome X. The two halves are held together at the centromere, in the center unit, which is a group of proteins in a restricted portion of the chromosome.
Okay, so I would sketch this out if you didn't already. You've got one linear strand of DNA. What do we call that? Chromosome. You make a copy of it, and you hook it together in the middle. What's the part in the middle called? We just went over it. Centromere. Okay, so it's hooked together right there. Each one of these is individual, or what are they called? I mean, just each one, the pairing. Sorry. Sister chromatids. Okay, and then this entire structure is what? It's still just a chromosome. Okay? So, everybody understand? Yes? Good? Okay. Super. You people out there in the internet world understand? Super. Okay, mitosis versus meiosis. Huge shout out to Mr. Armstrong for getting me this super cool looking tripod. And thanks to Mr. Menon for letting me steal one of his iPads. Mitosis, ordinary cell division. What's mitosis? What's it for, Miles? Yes, to make identical copy cells, either for growth, repair, division, any of those things. Mitosis to make two identical cells to the original cell. Meiosis is for the production of gametes. The difference between the two is mitosis, you should have the same DNA at the end. Meiosis, you should have half the DNA, and every sperm and egg and sex cell you have should all be different based on how those are split apart. But like I said, we'll get into that a little bit later. All right, turn to part 2.
Mr. Knight, you said the thing in the middle, that's called what?
Centromere. Do you have the notes?
Yeah.
Yummy.
Oh, never mind.
Good job, Mom. All right, if you'll turn back to 236, and y'all should have drawn this out the other day, hopefully. Maybe. Bless you. All right, let's see here. Important concepts. Centrioles are components of the cytoskeleton that are composed of microtubules. Centrioles are important. They will help in division. Microtubules are also compose the spindle fibers of mitosis and meiosis. So what you're going to find here, and what we've talked about a lot during the cell units, is that the cell is really good at recycling stuff. The cell is like super duper recycler, so it can turn one thing into another thing and reuse it for this, and when one thing wears out, it can reuse those components. So, good job, cell. That's a way to be environmentally conscious. Proteins are the workhorse of the cell. That's kind of a weird thing to say. Basically, they help things move and reactions take place and all that kind of stuff. All right. The cell cycle phases of eukaryotic cells. So, on page 236, your book breaks this down. I know it splits prophase in two. We'll talk a little bit about what that means and why they have that. Your notes really doesn't do that, but that's okay. So, most of the time, cells are in interphase. Now, how much time they spend in interphase is dependent on what type of cell they are, where they're located, how often they have to divide. Some cells may spend all of their life in interphase after after you're born. Most of your muscle cells and neurons pretty much have the same ones throughout the course of your life. Some cells, like your skin cells or stomach cells, they're going to be constantly dividing throughout the course of your life. Okay? All right. Just really depends on what type of cell it is. Megan, read part A for us.
Metaphase cells spend 90% of their existence in this phase. This phase consists of 3 parts. G1, primary and first growth. This is ordinary everyday activity to repair the cell. Animals begin with replicating. This checkpoint, called ornamental term, is the period to the rest of the cycle.
Okay, hold on there. All right, turn back to 235 real quick. All right, if you're in G1, if you look on, okay, normally we refer to G's as the G phases as growth phases, because really that's what's happening a lot here. If you read through 235 in your book, it talks about, at the bottom left there, the G phases were misnamed as gaps when they were first observed because the cells appeared inactive, but now we know that intense metabolic activity and growth is occurring throughout interphase. So there's no real gap here. All 3 subphases. Now, there is a G0 phase that we'll get into. Actually, if you skip on down in your notes there to part D, G0, zero growth phase. Cells are tired and take a brief break or rest, or they stop adult development. On 244, let's just go ahead and look at that. So let's pretend the cell has stopped dividing, and on the bottom right of 244, if you look at G0 there, if it does not receive a go-ahead signal at that point, it may exit the cell cycle, switching into a non-dividing state called G0. Most cells of the human body are actually in the G0 phase, as mentioned earlier. Mature nerve cells and muscle cells never go back to divide. Other cells, such as liver cells, can be called back from the G0 phase to the cell cycle by external cues such as growth factors released during injury. So, a lot of cells in your body are in that G0 phase where basically they're in a non-dividing phase. We good with that? I like y'all's enthusiasm this morning. Siri, stop listening to me!
Sorry, I can't do that.
AI is going to take over the world.
The FBI agent.
I had to kill Alexa at my home. Not my daughter, the machine. Okay. Let's see. Where do we get to? Okay. So G1 is our first initial growth. What's happened right before G1, Lauren? Yeah. So the cell just got through dividing. So what's it probably going to be doing there? It's going to be growing. Yeah, it's going to be maturing. Organelles replicating, growing, maturing, getting to its normal, big, functioning state, whatever that is. Okay. S phase, we talked about this the other day. Read that for us, Devin.
I think my notes, but I still don't know the first page.
You should get those. Carter. We're at the next one.
The DNA replicates or synthesized during this phase. In humans, we go from 46 chromosomes 2N to 46 chromosomes 2N plus the replicates. Still consider 46 chromosomes even with replicates.
Yeah, sometimes they refer to it as 96, which I think is just bad and confusing. Basically what's going on here is you've got your 46 chromosomes, and then they have a duplicate of each one of those, okay? A duplicate copy. And then G2, secondary growth. Read that one for us, Jordan.
The organelles mainly enlarge or complete replication. The newly synthesized DNA is checked for errors. Second checkpoint occurs after this part. Do we have everything for 2 cells? If yes, then proceed to dividing. If no, then make what is missing.
Okay, so you've seen these checkpoints listed here in G1 and also in G2. Why does the cell cycle need checkpoints?
Well, if they make one thing wrong, then every cell after that's going to be wrong.
Okay, so the checkpoint is to make sure that everything is there that's supposed to be before it proceeds on to the next stage of development, because you don't want some malfunction with that cell. And we talked about how sometimes, you have cells in your body that are dividing every day or two, and you don't want mutations to happen and to get passed on. So if you guys will turn to 243 real quick. On 243, this shows you the checkpoints at the top, kind of an overview here. Mechanical analogy, blah, blah. Okay, so you can see there's a checkpoint toward the end of G1, okay? And then there's a checkpoint at the end of G2 before mitosis occurs. And then there's another checkpoint during mitosis, which we'll get into in a second, okay? So these are in place in the cell to make sure everything is dividing and going along properly. All right, mitosis. Mitosis is specifically referring to what? Nuclear division, yes. Okay, so if you look on 235, let's go back there real quick. It refers to this whole division thing as M phase, mitotic phase. But that also, M phase includes cytokinesis. Mitosis is technically the division of the nucleus. Cytokinesis is the division of what? The cytoplasm. The latter part of mitosis and cytokinesis are happening concurrently. So I know it's got them drawn as two different things there, but really there's overlap. So skip over to 236, or back to 236. Mitosis. All right, PMAT, however you want to remember this. Whatever works for you. You need to know the order. You need to know what's happening in detail in each phase. I would imagine that probably you will have a written component for this on your test. Prophase. Who wants to read that one? Go, Ivy.
Nuclear envelope is broken down and rearranged to make the spindle apparatus.
quite a few things are happening there in prophase. Now, tomorrow when we get the microscopes out and we look at these, we're going to have to, well, hold on, tomorrow's Patriot Games, isn't it? Gosh, don't get me started, I'm on camera. Whenever we get the microscopes out and start looking at these, you guys are going to have to go kind of quickly because, well, I guess maybe, is anybody in Patriot Games in here? Okay, well, maybe we can get started tomorrow. So, the nuclear envelope's broken down. If you're going to divide the nucleus, you got to break down the nuclear envelope. Chromatin condenses to form chromosomes, so you got to make sure all your DNA is coiled up into chromosomal form. Those little characteristic X shapes, even though they're not really X's, they're more like back-to-back chromosomes that are hooked together in the middle. And then centrioles move toward the poles in animal cells. Centrioles are what? Am I remember? I don't think it. Let's just look in the book then on 236. See these centrioles? They're all part of this centrosome aster complex here. They're kind of the things in the middle. Does anybody remember what centrioles are? Yes, so centrioles are anchoring points for the spindle fibers. Basically what they're doing is like, so you have an animal cell that's a little squishy and not super rigid like a plant cell or any kind of cell with a cell wall. And so if you're going to pull something apart in the middle, you have to have anchoring points at each side. And so the centrioles, they're going to move to opposite sides, and they're going to act as those anchoring points for the spindle fibers to help move the chromosomes apart. Okay? So that's important. Let's see what else. Really quick, let's just look on interphase there at the bottom. Centrosomes, I'm on the third bullet down on the left side. Centrosomes are regions in animal cells that organize microtubules of the spindle. Each centrosome contains 2 centrioles. So they're the areas, the centrosome has the centrioles and where the spindle fiber is attached to, basically. Okay? So if you see that term, centrosome, that's what that is. Okay? Not to be confused with, what do we call the place where the sister chromatids are attached? Centromere. I would make sure you write these down and know the difference because they're all kind of similar. The asters there, it talks about them on 236. I don't know that I've seen that on the test, but I think maybe one time, a long time ago. That's where the centrosomes and the spindle fibers all kind of radiate outward. I think that means stars. Anyway, I don't think you'll see that. So, that's what's going on in prophase. Now, your book does what to prophase? It splits it up. So it says early prophase, which is where you can start to see the chromosomes. The nucleus is starting to break down. And then pro-metaphase. Obviously, this whole cycle is not like one step, another step, another step, another step. It's on a continuum as the chromosomes are condensing and moving toward the middle and then separating and then moving apart. There's an early and a late phase to all these. Really, pro-metaphase is when you really start to see those chromosomes and they're all kind of migrating closer and closer to the midplate there. Okay, they become a lot more evident. They've got an early prophase drawn here where those chromosomes have just started to condense. But really the spindle fibers and everything hasn't separated and the spindle fibers haven't hooked up yet. Okay, but like I said, it is a continuum. You could probably guess which phase do you think would take the longest here out of all these? Probably pro, because there's a lot going on there, right? That's actually one thing we're going to test for in the lab. So you guys are going to be counting how many cells are in each stage of the cell cycle, and depending on how many cells out of a large grouping are in each stage, you should be able to determine how long they spend in each stage of the cell cycle. So if you know a cell, like we're going to look at onion cells, takes 1 day to go through the complete cell cycle, if you know how many cells are in each stage, you should be able to determine mathematically like how long each stage takes. What's up, so Devin? All right, metaphase. Who wants to read that one for us? Good job, Hunter.
Meta means middle. All chromosomes line up on the metaphase plate, which is the middle of the cell. The spindle apparatus attaches to the kinetochore.
There you go.
A part of the centromere and the centrioles, the anchors. Third checkpoint occurs here. Are all the chromosomes attached and lined up and ready to divide/separate or segregate?
Okay, so here is our other checkpoint. During metaphase, do we have all the chromosomes? Are they all lined up in the middle properly? You'll notice on the metaphase part there, it's really, they've got it on pro-meta and the metaphase part. You can see these kinetochore structures that are forming, which is basically where the spindle fibers are attaching to the centromere. Okay? Third checkpoint, we got everything. Is it all lined up? Are we ready to separate those chromosomes? That usually happens pretty quickly compared to prophase. Anaphase. Paulina, you read that one?
Anaphase, ana means separate. Chromosome pairs are pulled apart, each chromosome moves toward opposite poles or ends of the strap. The spindle apparatus is being broken down as the two sister chromosomes are walked toward the pole by the mother protein using ATP.
Okay, so anaphase, remember from 9th grade, is where the chromosomes separate, they're moved toward opposite poles. The note writer makes note that they are walked toward the poles using motor proteins, and it does require energy of ATP. Okay, so what do they mean by walk toward the poles? Hold on a second. We'll get into that in a second. There's an experiment we'll look at. All right, and then telophase, the last part here. So telophase and cytokinesis you would expect to be occurring concurrently with one another. So the cytoplasm is being split into and telophase is taking place at the same time. Does that make sense? So I know we usually write it like telophase, cytokinesis, but they're really happening concurrently with one another. All right, telophase, read that for us, Brianna.
Telo means last. The nuclear envelope is rebuilt by using broken down spindle apparatus pieces. The chromosomes being to decondense back to their chromatin site. A cleavage furrow begins to form using actin and myosin microfilaments.
Yeah, for those of y'all don't remember, actin and myosin is what is used in like muscles, and we'll look into muscle contractions later on. Chromosomes can become decondensed back to their normal state, and those spindle fibers, remember, they were made from what? In prophase, they were made from. Hope you people at home are paying attention. What? The nuclear envelope. If you look back at prophase, nuclear envelope is broken down and rearranged to make spindle apparatus. In telophase, nuclear envelope is rebuilt using broken down spindle apparatus pieces. So they are recycling and reusing those for nuclear envelope and for the spindle apparatus. Okay? So telophase, you start to have two new nuclear envelopes forming. Chromosomes start to decondense again, so they can be that information can be used. And then while that's going on, then the cell is also getting squeezed into. So these are things you're going to be looking for in the cell on, I don't know, whenever, hopefully tomorrow. All right, cytokinesis. Cleavage means a split or a break. This is division of the cytoplasm. Cytoplasm and organelles are separated to produce two daughter cells. And you can see that toward the end there. Now, in plant cells, that is significantly different. Why? Because of the cell wall. So if you look over, let's skip down real quick to where it says cell plate. Rusty cell plate? Cell plate, cell plate. Remember, plant cells do not have centrioles because they have cell walls to anchor to. So they don't need centrioles for anchoring points. The new cell wall in the middle will become basically what's called a cell plate develops. And so the cell plate are just basically little segments of cellulose that start to be laid down the middle of the plant cell. There's a picture of it on page 241 at the top right side. If you look at 241 top right side, this is what you'll be looking for because we'll be using and plant cells, yes, primarily in the lab. And so when you get to this latter part of telophase cytokinesis, you should see this little cell plate, this line forming across through there, which will become the new cell wall. Okay, so obviously a plant cell isn't going to like squeeze in like an animal cell is. What did I skip? We talked about G0 already. Spindle apparatus. So, spindle apparatus, why is it important? How does it work? Turn to page 239 in your books. All right, spindle apparatus. Read A and B for us, Miles.
These structures are formed from the broken down cytoskeleton and nuclear envelope. They are recycled. The construction starts at the centrosome, where the centrioles are, and works towards the chromosome.
All right, so we already talked about how they're recycled. Construction starts at the centrosome, works towards the chromosomes. Part C, they attach to the kinetochore of the centromere of the replicated chromosomes. So you can almost think of the, like if you take your hands, your palms of your hand would be like the centrioles, or really, I guess you could say centrosomes. And then your fingers would be like the spindle fibers. And where they attach, in the middle, they attach to those kinetochore structures, or to the centromere, which forms the kinetochore. Okay, and then those chromosomes are going to be walked along, almost like down a road, walked along the spindle fibers. And there's a good picture of how they figured this out on 239. It was thought like which one's moving, are the spindle fibers getting shorter, how are they getting pulled apart? So they did an experiment to figure this out. Let's see, did I miss anything? The motor protein walks the sister chromatid toward the opposite poles by using ATP by phosphorylation. Non-kinetochore spindles are used to push the poles farther apart to help produce the cleavage furrow. So there's a lot going on here. Let's look at the experiment on 239. Read the experiment part for us, Isabella, at the top.
Gary Borsi and colleagues at the University of Wisconsin wanted to determine whether kinetochore microtubules depolymerize at the kinetochore end or the pole end as chromosomes move towards the poles during mitosis. First, they labeled the microtubules of a pig kidney cell in early anaphase with a yellow fluorescent bag.
All right, so what they're trying to figure out is which one's getting shorter. Is it the end toward the next to the chromosome, or is the end near the centriole? Read that next paragraph for us, Devin. Then they marked 239. Second thing down from the experiment. You gotta wake up. Lauren, read that for us.
And they marked the region of the kinetochore microtubules between one spindle pole and the chromosome by using a laser to eliminate the fluorescence from the region while leaving the microtubules intact. As anaphase proceeded, they monitored the changes in microtubule length on either side of the mark.
Okay, so what they're doing there is they're marking it, and that'll give them a reference point. Basically, if they mark it down the middle, they can see which side of that mark's getting shorter. So how is it getting pulled apart? Okay, results, Abigail.
As the chromosomes move forward, the microtubule segments on the kinetochore side of the mark shortened, while those on the spindle pole side stayed the same length.
Okay, what's our conclusion then, Angela?
During anaphase in this cell type, chromosome movement is correlated with kinetochore microtubules shortening at their kinetochore ends and not at their spindle pole ends. This experiment supports the hypothesis that during anaphase, the chromosome is walked along a microtubule as the microtubule depolymerizes at its kinetochore end, releasing tubulin subunits.
Okay, what's that big fancy word, depolymerize? What's a polymer? A bunch of monomers hooked together, in this case, to make the spindle fiber. So to depolymerize means it's what? It's just breaking down. So now we know which end it's breaking down on. As it's walked toward the centrioles, okay, on the opposite poles of the cell, it's breaking that down as it goes along, okay, but it's not broken down toward the centriole side. All right.
I'm
Now,
sorry.
started doing that yesterday. I don't know. It's like she went crazy. Telling you. Yeah, it's coming for you. All right, 240. Cleavage furrow. That looks like a very strange looking balloon or something there. I don't know what you would call that. But you can see how the cleavage furrow is starting to cut into the cell and where it's going to be splitting apart. And then at the bottom of 240, you can see the cell plate formation where new cellulose fibers are being laid down, which will form the new cell wall. And then regulation. What does regulation mean? Control, thank you. You've been studying politics, you know this, right, Abigail? Classic. Regulation, control of the cell cycle. Regulation is crucial for normal cell growth and development. Regulation varies from each type of cell. What happens if you don't have control of the cell cycle?
Then it gets out.
Which we call what? Cancer. Good. Yummy. All right, where is some...
I don't know if that was the right.
Yeah.
Can I edit that out? Okay, it's not yummy. Let's see here. Regulation. Regulated by proteins called cyclins. Let's flip over to 243. Actually, 244. We already looked at the checkpoints, right? Okay. On 244. Molecular mechanisms that help regulate the cell cycle. I would be familiar with these. 3 checkpoints exist. We already talked about those. The one at the end of G1 before it goes into synthesis. The one at the end of G2 before it goes into mitosis. And the one during metaphase to make sure you've got all the chromosomes lined up. Everything's in the middle. Everything's there before it splits. Now, does that mean the cell doesn't mess up its exchange of DNA when it's dividing? What?
Yeah, like, yeah, it'll still mess up.
It can still mess up. What's an example?
I was gonna say something like that.
Yes, Down syndrome is probably the most classic example where the chromosomes don't separate properly. And there's a number of other ones. We just hear about Down syndrome a lot because it's probably the most common lived with, but there's all kind of chromosomal disorders where chromosomes do not separate properly.
Is that like Klinefelter's syndrome?
Klinefelter's is another one. Turner's, where you don't have a proper pulling apart of the chromosome. Now everything is probably there and correct up to that point, but for some reason they're not separating properly. Okay. What's the difference between? So the last part of the notes here. What's the difference between theta chromosome and eukaryotic chromosome? Theta means what? Circle. More genes are present in eukaryotic chromosomes than on theta chromosomes. It varies wildly from organism to organism, but bacteria tend to have way fewer genes, chromosomal material. Although I will say this, what we know about bacterial DNA, almost all of their DNA is coding for something. A large portion of our DNA is non-coding. The last thing I saw from HMI, I think, was maybe like 2 or 4% actually codes for proteins in our DNA. We've got a ton of like pseudo DNA that no longer codes for stuff, old viral DNA that doesn't code for anything, a ton of stuff that has to be cut out. So anyway, whatever they have, they're using it well. There exists more genetic stability in eukaryotic chromosomes. Offspring receive the same number of chromosomes most of the time. Important linked genes tend to be inherited together. We'll talk about that when we get to genetics. And this allows for diploid cells to exist. And diploid cells meaning you can have undergo sexual reproduction, which allows for more variation, which allows you to take over and conquer the earth, Carter. Am I right?
Yeah.
Okay. So let's go through. Let's stop there. Let's take a quiz.
Wait a minute.
All right. We'll go over part 3 tomorrow, and hopefully we'll start the lab. That's my hope and dream. We'll see how that works out.